how this Facebook actually find or identify a face of your friend from a billions or millions of different photographs. And that is the answer is the artificial neural network. In a Facebook server, the artificial neural network they use, it probably it has somewhere about 50,000 different artificial neurons in that artificial neural networks. Now, in the most of the cases, again, 98% of the cases, this artificial neural network is implemented to softwares, those are the computers, right? But as I said, if you want to make a real robotics, you cannot just connect a remote server to a robot. That's not a good idea. So if you want to make a robot intelligent, you have to actually do a hardware implementation of the ANN, not a software implementation. So that's why there is one research field, and this is who developed a lot of different kind of neuromorphic chip, so that the whole ANN can be performed in the chip only. So then put in the chip. So that is basically a lot of inorganic material-based chip people develop. People actually do that ANN through the photonics. And people showed that it can be done in the in vitro DNA computation. Means they can in a test tube, giving just the DNA into it. What is my lab is interested, or the question we do ask, that can you make a bacteria as an artificial neuron, and then a population of the bacteria can work as an artificial neural network which is made by this bacteria. And to do that, we chose something, a decoder encoder. I'm sure that most of the engineering students, you know what is the decoder encoder, but who doesn't know, just to give a brief introduction. So this decoder encoder are always used in a lot of wireless communication system. For example, if there's a walkie-talkie, and this walkie-talkie, when I'm talking, then this sound signal goes to the electrical signal. That electrical signal is first got encoded through a circuit or device called encoder, and that encoded to a fewer bits, and that information is sent to the other instrument, and other receiver actually receive that, and that's encoded signals actually first decoded through a decoder, and then actually come to as a sound signal, and you can hear that. So there's a lot of communication system in the avionics industry and so many things. What I'm just trying to say, that the decoder encoder is a very important system, and what we would like to do this decoder and implement decoder and encoder using bacterial artificial neuron. So I'm just trying to give you showing the result. So this is an 2 to 4 decoder. This is the truth table, and this is the X and Y at the input. And based on the combination of the input, it has 4 different outputs. And if you can see here that diagonally there is 1, 1, 1, everywhere is a 0. So what we make this system using bacteria, but not following this electronic engineering principle, but just following this artificial neural network way. And I'm not going how we're doing that, but there's a lot of molecular engineering is there. And then we feed these equations, a lot of mathematics to change that, how this weight and bias is going to be adjusted, and so on, so on. And finally, when you actually make this network, and this is an experimental result, you can see diagonally there is only fluorescence like red, purple, green, yellow, and everywhere it is black. So exactly it is like that. Diagonal is 1, 1, 1, everywhere it is 0. So we're excited because you can make this 2 to 4 decoder with the bacteria using ANN. We have done encoder again. I'm not going. This is a truth table. You can see this is much more complex than the decoder. So this is our ANN with our artificial bacterial neurons. Because remember, this is in hardware, because it is actually happening. It's not a software. It's not the mathematics. This mathematics is actually happening with the bacteria. And then when you do this kind of experiment, you get the results. And if you match them, they are actually the same. So we also try to do something, right? You probably heard, I don't know about this, anything about the quantum computer, okay? You may know much more than me, but just what you say, that in quantum computing, there's the platform of the, if you want to do through logic gate kind of a based platform, then the irreversible computing, what we do in AND gate, NOR gate, NAND gate, it is not the way. So you have to do through reversible computing. And this reversible computing has 2 different aspect. One is thermodynamic reversibility, another is logical reversibility. Now, what you have studied in the thermodynamics in your chemistry or the physics class, specifically in your first or second years, that thermodynamics, specifically the reversible thermodynamics, that is not applied to the biology, right? So when I said we have a reaction A plus B goes to C and it ends, right? But in the biology, the reaction doesn't end. This is I'm talking, you are listening, or you're pretending listening, probably you are I don't know, sleeping, okay, or whatever you're doing. So all these out of the equilibrium, right? You cannot do the reaction doesn't stop in the biology. If the reaction stops, means I dead, right? I'm dead, but we are not dead. So we are out of the equilibrium. So the whatever the thermodynamics what has been used in a system is basically irreversible thermodynamics, and people use this the thermodynamics in basically in the chemical engineering in an engineering field, and the whole biology is irreversible thermodynamics. So however, therefore we cannot attain thermodynamic reversibility if you want to make through this bacteria ANN. But what we can attain, what we call is a logical reversibility. And let's see what is the logical reversibility is. Now here, main thing of the logical reversibility that it has the information get preserved. So the input looks exactly like the output. So this is a Feynman gate, or Richard Feynman actually proposed this gate, and it is also sometimes called controlled NOT gate. So now if you see, if it was a kind of AND gate, you will see only when there is a 1, 1, 1, then your output is 1. Everywhere it is 0. So information is not preserved for the other 3 cases. But here you can see that A and B, if it is 0, 0, P and Q output is also 0, 0. If it is 0, 1, it is 0, 1. If it is 1, 0, it looks here 1, 1. Definitely the output is for this particular it is not preserved in straight way, but in a zigzag way it is preserved. So this 1, 0 is come here 1, 0, and this 1, 1 is come 1, 1 here. So you can see that information is preserved. So what with these things we actually developed this Feynman gate with this bacteria ANN, and basically what you can find, this is the actual result here. Okay. So another area of our work, what we do, which is called the space synthetic biology. As I say, the synthetic biology could be the key for the future space travel. So Adam Merkin and their group actually proposed there are 6 areas of the future space technology where synthetic biology can have the contribution, and starting from resource utilization, manufacturing, life support, space medicine, space cybernetics, and terraforming. So in all of that, what is basically we need in some of the cases, the microgravity is an integral part of the system. So this microgravity what does, like when there is an astronaut goes to the space, that immune system got deregulated, as people know from the Apollo mission. At the very same time, our own gut bacteria, which is in our gut at this moment and doing nothing, no harm to us on the Earth, if we go to the space, our own gut bacteria got virulent. So this is a very clear combination. But people do not know that how microgravity affects these tiny particles or tiny bacteria or tiny cells to make such kind of the phenotype. People studied it very well in space and microgravity simulator on the Earth. However, somehow their statistical analysis technique is not that good. You cannot take a lot of information out there. So what we actually developed a systems biology pipeline using several statistical tools and machine learning algorithms, and we got several new insights what other people could not find from their own data, right? And then we published that. I'm not going into the detail. Just one thing I want to mention that here we used something a very different engineering, and we use something called non-negative matrix factorization. It is a machine learning algorithm, and specifically people use for the face recognition. The same machine learning algorithm we have used to actually analyze the space-related genome data. So this is interesting, right? How this engineering and biology are doing very well together. And when we actually publish those papers, just we got featured in Times of India, Nature India, some other newspapers, and so on. Okay, so whatever we got insight from there, our goal is to develop something called a microgravity sensing biochemical device. So we developed a synthetic gene circuits, and we actually tested in our microgravity simulator, which we have our lab, which is designed by the NASA, and you can see the result. When there is a microgravity, the signal is almost 0. When it's gravity, the signal is around 30. So there's almost 30 times difference in the signal. So we're happy that we can do that. And with the microgravity, we can control the cell shape and cell division process. Here is so in Earth's gravity, so we engineer the cell in such a way. In the Earth's gravity, you see the cells looks like some elongated way, right? Elongated. This is not the exactly cells thing. So now our device say that when you actually put in the microgravity, this shapes has been cured, and now you won't see this kind of elongated shapes. So we're very happy that we were able to make world's first microgravity biosensor, and we can use this. We can use this to actually control many cellular processes with that. Now, this you can think about an engineering problem, right? Now think about the energy. Now we do need a lot of energy when you actually have the street lights. Now think about that amount of the light takes a lot of amount of the energy and lot of the cost. What about you can actually develop this kind of the solution to the energy? This is taken from the movie Avatar. So if you make the tree, whatever's happened just in the roadside tree, they glow like this, then you do not need those kind of electrical things. So this is another way to think about the solution, engineering solution for a lot of this problem. And with that, I like to stop here. And this is my small group. They're all my PhD students, and he's the scientific assistant. And this is the funding from where we got to do our research. And thank you very much for listening.
Thank you, sir. Thank you very much. It was indeed motivating, enlightening, informative, and very interesting. I think the session is now open for questions, sir.
Yes, please.
Okay, so I request all the participants to please come forward with their questions. They can also write it in the chat box and I'll read it out for them. Do the participants have any questions? So please, you can come forward with your questions.
Students can put their questions on chat box also.
Yeah, I request them to write their questions in the chat box, but still in the chat box I don't see any question.
Sir, one thing I'd like to ask.
Yeah.
Hello.
Yes, yes.
So, how synthetic biology helps in today's biomedical instrumentation?
Biomedical. So see the okay. So the biomedical instrumentation. I mean, one of the important aspect of the biomedical instrumentation is basically the robotics and the AI. And I would show that with the synthetic biology, what you can develop a lot of biohybrid robots. So, for example, so let's say inside the human body, you want to see something, and now if you have an engineered organism like the bacteria you put through your bloodstream, this bloodstream, this bacteria can go at a very different spaces, and then actually if you have a disease state, like if there is a cancer is growing at some point, there is the microenvironment signature of that cancer, it will produce some chemicals, so that if this bacteria can actually identify that. So and this bacteria it can be full of your drugs inside it, so it can actually deliver those drugs there. So now instead of that, if you use a biohybrid, not just a bacteria, you can micro swimmers which is connected with the bacteria, so you can actually using a magnetic control, you can actually control the movement of this micro swimmers. But the bacteria will take all the biological, it actually take the biological information from it, and it will send to you through your whatever the micro swimmer. So this is the one area to actually go for the biomedical instrumentation. So the other area, I mean, the whole diagnostic platform is basically people are trying to put everything as a bacterial sensor. So I said there's the bacteria, but it's not need not be the bacteria. People actually developed a lot of mammalian cells, and what you can put in your, for example, in your brain. Right, and this brain, it is an optogenetically engineered, which basically means that you can actually control that the cell function by applying light from the outside, right? And those are the things you can actually do that. There may be other also. I don't know all of them, but this is just a few things.
Okay, thank you.
Yeah.
Thank you, sir. We have a question here from Polashnath Das. He is
Yes.
asking whether we can make solar panels using human hair.
Okay, so I really don't know that if it can be done. I mean, I never thought about in that range. But what I can give you information for so people